Great pleasure to be here in the Science Gallery as usual. Who won the Shorty Award, is that right?
Yes.
What is the Shorty Award? I'm not Shorty. But it's a great achievement. Twitter gave out this award and the Science Gallery won it, and it's wonderful to see that. And I'm a huge fan of the Science Gallery, needless to say. I'm also a huge fan of SFI because they gave me, or have given me, €6.6 million. Right? Round of applause for SFI. Okay. Now, that sounds like a lot. It's not really in my game, you know. In immunology, very expensive business. We spend about $1 million a year on my research program, and you need that kind of money to compete internationally. But it's great that SFI are here in Ireland to give us that money, and long may it continue. Now, I'm going to really give you an hypothesis to begin with. I am going to use some jargon, terrifyingly. Around about 10 jargon words, which I want you all to remember. For instance, NLRP3, there's a terrifying word. PML is another one. I'll come back to those later. About 10 words of jargon. But I'm going to hypothesize that if we understand the immune system, we will find cures for cancer, rheumatoid arthritis, multiple sclerosis, asthma, malaria, TB, all the major diseases that afflict humanity will be cured through the immune system. And that's no proud boast. We really believe that in immunology. So imagine for a minute a world where there's no cancer. Imagine that. Or no atherosclerosis, heart disease, or no arthritis, or no MS, or no motor neuron disease. That's the promise of immunology, and that's what we're going to try to discuss tonight. And I'm not saying that as a press thing. You know, in the Irish Times, ONeill discovers cure for cancer. We really believe this because we have the immune system in our sights. We know how it works in great detail. We're learning more and more every day. And my research program, and many of us all over the world, are part of this big adventure to explain the immune system and from there into disease. So let's look at this for a minute. This is the problem that we confront, of course. Cancer, I got this morning on the web, 7.6 million deaths a year. Malaria, 2 million. And even diabetes, your life is shortened by 10 years. Arthritis, shortened by 5 to 10 years. So even though those two don't kill you, they shorten your lifespan because the stress your body is under when you have those two diseases is severe. Now, that's if anybody has diabetes, don't worry too much. This is about 3 or 4 years ago, and they are shortening that with the new treatments. But these can be lethal diseases and can shorten life. So death is the first problem. Whatever about the horribleness of having MS or motor neuron disease, death is another feature of these diseases. Now, and as I say, in the immune system, this will be our hypothesis, all of these involve the immune system dysfunction, and in particular, a process called inflammation. Now, inflammation is a very important part of our immune system. When that goes wrong, you get these diseases. That's what we've discovered in the past 5 or 10 years, I guess, especially. Now, inflammation and immunology are cool. And how do I know they're cool? I'll give you 2 pieces of evidence. First of all, this guy here. Do you know who he is? Bill Gates. The Financial Times interviewed him about 3 months ago on a Saturday. They interviewed someone and have lunch with them. And look what he says. Immunology is super interesting. So we're all doomed now if he thinks it's interesting. Because he is the uber geek, isn't he? But he really thinks immunology is fantastic. And he said this in a great speech he made in Harvard. He said, humanity's greatest advances are not in the discoveries, but in how those discoveries are applied to decrease inequity. And he was talking about malaria and TB and HIV. The biggest inequity of all is in health. The reason why people die of TB and HIV is mainly poverty. So Bill wants to cure those diseases, to right the wrong of this imbalance in the world. Inequity will also apply for someone with cancer. That can be another economic problem, or just bad luck. I mean, people get these diseases, and there's an unequalness in the population. So really, immunology is the way, is what Bill says as well, to try to get at these. The second reason it's cool is, look at this. In December, Science magazine published a really interesting issue. They called it Insights of the Decade. And they listed the 10 big science insights of the past decade, one of which was inflammation. And they said over the past decade, it has become widely accepted that inflammation is the driving force behind chronic diseases. Most diseases I talk about are chronic, and it's become clear it's dysregulation in this process that drives these diseases. And really, why would that be? Why would the immune system and inflammation be at the heart of all these diseases? Well, really, in many ways, you can define disease like this. It's displacement from homeostasis. As you all sit here, you're all healthy. Your body is in a homeostatic state. Everything's balanced. You know, your blood's flowing right, your heart's beating, your kidney, your brain are working. Everything's fine. When you get disease, it's displacement from homeostasis. And the job of the immune system, very, very simply, and inflammation, is to sense that displacement and restore it. That's what it's all about. The best example is an infection. So when your body has TB, you're displaced from homeostasis. Your immune system senses that displacement and then deals with it. And finally, hopefully, gets rid of the pathogen. And in the case of a horrible disease, will ultimately treat it, hopefully, in your own body. So this displacement is the key concept here, I guess, as to what the immune system is doing. Now, look at this next thing here. So My lab every year, anybody from my lab here? They're all too frightened. Oh, there is. Last Christmas, they gave me a present, and this was the annual present. They gave me this book, which was very funny, right? You know when you're in an exam and you don't know the answer, and they're all students here, and you spoof the answer in the hope of getting a good mark? This book is the best description of all the best spoofs ever. And look at this one. In biology, explain the concept of homeostasis. It is when you stay at home all day and don't go out. That's what homeostasis is. Another favourite I found from this book was this. This is another process. In the Hawaiian Islands, there are around 500 different species of fruit fly. Give a reason, there are approximately 500 varieties of fruit. Full marks for that. So, never trust students. And we see this all the time. It's like, don't we, Aoife? We're in agony seeing student answers. But this concept of homeostasis and displacement is where we're at, right, with this inflammatory process. Now, what is inflammation? This is inflammation here. Chilblains, horrible. You're all familiar with the inflammatory process. This guy, Celsus, was the first to define inflammation. He was a Roman physician in the second 25 AD, 50. He wrote a medical textbook, and that's the first ever account of inflammation. And he said there were 4 cardinal signs of inflammation. It's defined by 4 features. And in the good old days, when Bill Clinton was president, I used to use this to illustrate the 4 features of inflammation. Right? So there's redness here in Monica's cheek. There was heat being generated here. There was certainly pain, definitely swelling. But poor old Bill lost the function. But Celsus defined those 4 features. Inflammation is mainly vascular. The blood vessels open up, the blood rushes in. All the redness is because of blood rushing in. And the reason for that is to get the immune system in there. So an infection, you get inflammation, to bring all the blood in and all the immune cells go in and handle the infection, a really important part of fighting disease. Of course, when it goes wrong, we get these horrible diseases. The first sort of scientific attempt was this guy, John Hunter. I did my PhD in London in the Royal College of Surgeons. He was the founding surgeon of the Royal College of Surgeons. His most famous book is on inflammation, and it was published in the late 1700s. It was a treatise on inflammation and gunshot wounds, because he was funded by the army. As ever, funding was an issue back then, you know. And what he said is inflammation is not to be considered a disease, but it's a salutary operation, consequently to some violence or some disease. When it cannot accomplish that purpose, it does mischief. And that was the key thing that Hunter spotted. Inflammation is good. It's supposed to fix you. At that time, they thought inflammation was a disease in itself. He said, no, its job is to actually fix things. When it goes wrong, it does mischief, and this mischief is all these diseases that we now are talking about. Now, nothing happened medically for a long time after Hunter in terms of treatments. And this book here I found this book in a bookshop about 3 months ago. It's a medical textbook from 1914, right? Like a family medical textbook. And I bought it. It was like, 50 cents or something. And I looked up rheumatoid arthritis. If you were unfortunate enough to have rheumatoid arthritis in 1914, this is what they did to you, right? First of all, the diet ought to be as rich as can be digested. Consider amounts of meat, good sound wine or stout, and extracts of malt. That's how they treated you, right? Then it says, the best drug is syrup of iodide of iron, whatever the hell that is. And then it gets worse, right? It says, that doesn't work. Sea sand should be heated in a tin and poured over the affected joint as hot as can be borne. Now, not regular sand, sea sand. This is 1914, right? If that doesn't work, this is what they did to you next. Look at this. Another method which frequently does wonders consists of the application of large blisters over the spine, these blisters being kept open for a week or 10 days. This is rheumatoid arthritis. Right? And then it says, it is usual to give some opiate. I'll bet it is. That's why it works during the cure, but the results are so gratifying in many cases. Who needs Enbrel, all these fancy drugs? All you want is a few blisters down your back. Nonsense. Most of the time, doctors did harm up until the 1920s, really, and this just typifies the problem of the medical profession. Horrific, right? And why did that change? It changed because of SFI and organisations like it. Basic research was required, right? Basic research began to be used on the immune system and inflammation research, and that gave rise to where we are now. And there's some really good justifications for basic research. The best one I've seen so far is old Sarkozy. He made a speech to the Institute of Physics in Paris back in January to defend basic research, because they're going to put 50 billion or something, the French government allegedly, into research in the next 10 years. And he said this, look, he said, without basic research, there can be no applications. After all, electricity and the electric light bulb were not invented by incremental improvements in the candle, right? And that was a very good way to put it. We need basic research. And who knows where it might go? I mean, who would have thought you would have gone from that to that, you know? And it's very important to fund basic research. We might say for arthritis, without basic research to come in application, aspirin was the mainstay as an anti-inflammatory for a long time. The best drug for arthritis now is a drug called Enbrel. It's an antibody that neutralizes TNF, and TNF is a very important immune component. It's made in Clondalkin, interestingly, a place called Grange Castle. They make that drug there. It's owned by Pfizer. It made $4 billion in sales last year, that drug, and it's made in Ireland, which is a good sign. And that is so, it's a protein, it's an antibody, it's so different to this. You wouldn't have got from there to there without all this wonderful basic research. So we need this research to give us this enlightenment, I suppose, as to how these systems operate. And of course, the scale of the problem cannot be overemphasized. Drug companies love inflammation because they're so common. 25% incidence overall. There'll be people here with inflammatory diseases. It might be psoriasis, very common in Ireland. It might be diabetes, that's inflammatory. Colitis, Crohn's, MS, asthma, rheumatoid. Infectious diseases are caused by a dysfunctional inflammatory process. So all of these diseases involve the immune system and inflammation. There are newer drugs around, and Enbrel is an example, but there's still a major unmet need. We can't treat these diseases properly yet, especially things like MS or motor neuron disease. Arthritis is a bit better. The newer drugs are working a bit better, but still a big need there. And look at this, cancer, very importantly. We're all convinced in immunology the cure for cancer will come from getting the immune system to attack the tumour. So as you sit here, this is a true fact. One tumour cell arises in your body per hour, everybody, right? And the immune system kills it. The immune system is always keeping tumour cells at bay. Can you imagine if we could understand that? And then if you had a tumour, you could mobilise the immune system. I'm going to give one example of that a bit later from my own research about how cancer could well be treated, I guess, through activating the immune system. And let me give you just a few examples of cancer immunotherapy, because there really is optimism here with this, that activating the immune system will treat cancers. Look at this, though. $90 billion has been spent on research since 1971 in the US on cancer. $90 billion. And we don't have a cure. There is progress, of course. We're still working hard. There are some leukemias can be treated. The incidence is going down. But these agents here, just a very brief list of them, these are all activating the immune system. So last week, the first drug ever to be approved for melanoma is a drug that activates T lymphocytes. And it was approved last week by the FDA. It's called ipilimumab, horrible name. They're using viruses to target tumours. They're using antibodies. You may have heard of Herceptin. That's used for breast cancer. That actually binds to a protein on breast tumour cells, and the antibody then kills the tumour cell as if it was an infection. So the view is out of cancer will come, or out of immunology rather, will come new treatments for cancer. Now, let me give you one example from my own lab. And this was done by a fourth year, a senior sophomore student. Is Gavin here? No, it's not you, Claire, sorry. It's not you. It's not you. I'm going to show results generated in the past 3 months in my lab by a fourth year undergraduate. And it's a really wonderful little study, this. Now, of course, it could be rubbish because he's a fourth year student. But all credit to Gav and a story in cancer. Right now, this story is a good one, I think, because it shows the power of technology. And in particular, and this is to give you wonderful lay people an idea of what we do in the lab, we use a technique called confocal microscopy in this study of cancer. It's a cancer project. Confocal microscopy was used. And what confocal microscopy does is you can see proteins in cells using this method. Individual proteins can be seen inside cells. And we looked at proteins in cancer. And of course, this technology was invented in a very strange way. The Nobel Prize for medicine was won for this technology. It's a way to look at proteins in living organisms and living cells. And it began with the jellyfish. Here's a jellyfish here. And these are fluorescent jellyfish, right? Begins with this in the 60s. And these three guys win the Nobel Prize for this discovery. This guy in the middle, Shimamura, he purified the protein from that jellyfish that fluoresces. And it fluoresces green. He called it green fluorescent protein, GFP. And he went about his business, became a taxi driver, strangely, right? That's all he did, purify GFP, and then he left science, right? This is true. When he won the Nobel Prize, he was in a taxi, driving his taxi cab. And the guy rang, "You've won the Nobel Prize. For what? A jellyfish protein? Don't be preposterous," he said. And what had happened was this guy, Chalfie, he figured, look, if we can attach this GFP to any protein, we can track that protein, because it'll fluoresce, right? And he cloned the GFP gene, and you can make these fusions between two genes now, and your favorite gene, it could be for any protein you want to work on, is now going to fluoresce. And it was a way to track proteins, right? And very importantly, Chen, he understood all the chemistry, figured out the chemistry of this fluorescent substance. He made variants by mutating the protein, and you get different colors, and that became very useful. And there is GFP fluorescing. This is the fluorophore in the middle. And you can make flies green by simply a single protein in that fly was modified all over the body, and now they fluoresce green. You can even make green rabbits. This is a real picture. Again, by tagging a common protein in skin, in rabbit, and that skin now fluoresces green. So it's a wonderful technology for tracking proteins. We use this, and there's the range of colors you can get, and you can tag proteins with different colors. And there's a confocal microscope from our lab, funded by SFI. They cost about €500,000, those machines, to give you an idea. And you can use them to image confocal proteins. And this is the project, very simply, right? There are two proteins of interest here. One is called PML, the other one's called ASK, jargon already. They're the two proteins. Why do we want to look at those two proteins? Well, PML is a very important protein. It's in leukemia, PML. It stands for promyelocytic leukemia protein. And in particular, it's not clear how it causes leukemia, but it kills cells normally, right, when they become cancerous. And the mutant, which is modified in some way, cannot kill the cancer cell. So mutations in PML are a cause of cancer. And we wondered, would it interact with a protein called ASK? Now, we got that by a fishing expedition. It was random. We isolated ASK, and then we found PML with it almost by accident. Now, ASK is in the immune system. It kills cells during infection, actually. So a virally infected cell will die when ASK is activated. And we found PML and ASK. We asked, would they interact? And would PML's capacity to kill cells go through ASK? And that was our hypothesis. Perhaps PML interferes with ASK. The mutant mightn't work, and then you might, get a tumor as a result of these proteins not working. This is the result that Gav got. That's what a cell looks like down a regular microscope. Horrible, right? Transfect that cell with PML. Now we can see the PML proteins, and we've got a red fluorescent tag on them. You can also stick in the gene for ASK, and it fluoresces green. See that? And you can also stain the nucleus of the cell with a special fluorescent dye called DAPI, and there's the nucleus there, right? And now we do what are called overlays. And first of all, if we look at PML and ASK, lo and behold, we get an orangey color. See that? That means this protein here is interacting with some of these proteins there, and now this orange color means they're interacting. That's confocal evidence. Now remember, I can't emphasize the wonder of this technique. There are 40,000 proteins in a cell. You can view PML on its own, or ASK on its own. That's why they won the Nobel Prize, really, because single proteins can be visualized. And this is the evidence they interact. And then also look at the nucleus. They're in the nucleus, and they're interacting in another color here by the three colors. Using confocal microscopy, we could show PML and ASK would interact. A fourth-year project student discovered that. Perhaps mutant PML cannot interact. So when it mutates, it can no longer interact. Remember, ASK will kill a cell. That's its job. The mutant is unable to kill cells, perhaps because it can't interact with ASK. And maybe if we can restore this effect, we could kill cancer cells. So there's an example of how you can use technology and, again, exploiting the immune system, because ASK's job was thought to be just in the immune system. Here's an example in leukemia, where PML through ASK might be very beneficial. And that's one little example. Now, the second example is malaria. So cancer, we've dealt with that now. We've cured that, right? Cancer will be cured through the immune system. Second example of malaria, will there be a vaccine? That's the big question. And of course, vaccines are the best contribution medicine ever made to humanity. It begins with smallpox, as you may know. A scourge on humanity, there's smallpox. Horrible virus, this, right? If I was to release smallpox in here, 1 in 3 of you would get smallpox. Of those 1 in 3, a third would die. A third will be disfigured horribly, and a third will fight it and get better. That's the funny thing about smallpox. It's in thirds, right? The vaccine, of course, was a really important thing, and the vaccine was partly discovered by a woman. You never hear about women in these histories. Lady Wortley Montagu, that's in our National Gallery, her painting. She was the wife of a Turk, the British ambassador to Turkey. She observed Turks sort of vaccinating in smallpox, but of course the key guy was Jenner. He used cowpox to vaccinate. And of course the trick here was cowpox is not as bad as smallpox, it's very similar. And sticking cowpox in, the immune system can then recognize cowpox. So because of the similarities with smallpox, it can also recognize smallpox, and therefore cowpox protects against smallpox. And the word vaccination is vacus from cow, because of smallpox. Now look at what vaccines have done for us. So here we have all these diseases. Right, let's take measles. In 1962, 500,000 people died of measles in the US. In 98, only 89 died because of vaccination. Right? Talk about a way to treat disease. And there's loads of diseases we can now vaccinate against, and the death rate went down to almost zero. They're the greatest contribution, really, as I say, of health, is getting vaccines to work. Big need for new vaccines. There's no vaccine for malaria, 1.2 million deaths a year. TB, 1.6 million. HIV, 0.8 million. So these are the big three that Gates funds. And of course, Gates is very keen on developing vaccines. And there's a great quote. They're going to put 10 billion in the next decade. Look at that. On vaccine research. Vaccines are the most cost-effective way to reach people and save lives, he says. And they're projecting all these lives to be saved if we can vaccinate against these diseases. Why can't we get a vaccine to these? We don't know enough about how the immune system handles them. This is the immune system on a single slide for you, to keep it simple. So here we have bacteria, virus, fungi, and so on. This is a key immune cell called a dendritic cell. That's the key sentinel cell of your immune system. This cell will sense these pathogens, and then you mount a response. The main thing that gets sensed is a thing called the antigen, which is a protein component in these bugs, and that gets taken up by dendritic cells. And we get T and B cells come out there, they're very important cells. They become memory cells. They're the ones that are that vaccines are trying to provoke. So cowpox will bring out T and B cells against cowpox and smallpox, and they become memory cells, meaning they're in your body for a long time. And then when you're exposed to smallpox, you remember that cowpox and fight the infection, and you're protected. And they're very important. And dendritic cells drive those. You also get antibodies, and very importantly, inflammatory mediators, IL-1 and TNF. Now, I mentioned TNF earlier. They are two very important signal proteins of the immune system. They kind of govern the entire immune response and drive inflammation. Now, that was the way immunology was up to about 12 years ago. And then a big revolution happened, and a whole set of sensors were discovered that can sense other microbial products, not just the antigen. And one family are called the toll-like receptors, or TLRs. They're very good at sensing bits of microbes. And they're required to get the dendritic cell to do its job, right? And that was a big breakthrough about 12 years ago that began. And that's my area. I work on these toll-like receptors. There are 10 of them in the human. They sense microbial products. Very importantly, they also sense inflamed tissue. So when a tissue is damaged, it releases material, and tolls also sense this inflamed tissue products. So in many ways, they're danger sensors. They sense infection and they sense injury. And what they do is they go through 4 proteins. These are called adapters. MyD88, MAL, TRIF, and TRAM. We were lucky enough to discover one of these, MAL, which is one of these key adapters. And everything happens in the immune system after this. The whole immune system is woken up by this pathway. And my own specialty within biochemistry is called signaling. We work on how signals are generated, and that's been my area for the past 25 years, how things work in terms of signals inside cells. This is a simple version of signaling, intensely complex. Now, we found this MAL thing as one component in this kind of mess. And then, very strangely, I am continuing a family tradition in signaling. You know, the 1901 census is online, right? And I searched for my ONeills, never knew about them because they were all thought to be convicts of various kinds, and the family wouldn't talk about them. I searched for the ONeills, and I found my great-grandfather, Daniel ONeill, was a station master. See that? And his son, Patrick, was a signal man on the railway. This is true. And I even found the station that Daniel was the station master. It's called Ballywillen in Longford. There it is. And there's no longer a railway line. There's Ballywillen, where my great-grandfather was the station master. His son, Patrick, my grandfather, was the signal man. And there's the signal box that Patrick used to. This is true. It's amazing. So I'm continuing a family tradition by working on signaling. Now, so here are the tolls. They're in the membrane. They're sensing all the nasties. They cause this alert reaction to happen. And let me give you a great story. This is the great one, right? 10 tolls in human. They've all been knocked out of mice, and you study the mice, right? This is really weird. About 3 months ago, this was published. One of them, number 5, they're numbered 1 to 10. Toll 5 senses a thing called flagellin. Now, flagellin is a key part of a bacteria, right? And in the gut especially, flagellin is in bacteria in the gut. And toll 5 senses flagellin and protects us in the gut from these. They made a toll 5 knockout mouse, expecting the mice to be all messed up in their guts and everything. And what they got was the mice became obese, amazingly, right? A toll 5 knockout mice gets fat. Even more compelling, they took bacteria from the colon of that knockout mouse and gave it to a recipient mouse, and that mouse got obese. Now just think about that for a minute. Look at this guy here. So I'll have the salad, thanks. The stomach's saying no, I want cheeseburger and fries. See? So the bacteria in your gut can make you obese. This is called the eat, shit, and die model of immunology. I'll move on. But isn't it amazing? So tolls, that's a little snapshot of how exciting tolls are. Tolls are governing bacteria in the gut. A second example is from our own work again, and we're very grateful again to SFI because they funded us to get to this stage. We have a company called Eczema, and we're developing an inhibitor of one of the tolls, toll number 2. And guess where number 2 counts? Luckily, it's not in the eat, shit, and die example. So this is rheumatoid arthritis. This is a rheumatoid biopsy from someone with very bad rheumatoid. You can take tissue from a rheumatoid patient, culture it. It pumps out lots of nasties. This is IL-6 here, TNF. We put our antibody into that ex vivo system of human rheumatoid. And look, blocked all the inflammation. Very nicely. There's TNF and IL-6. Now remember, the TNF inhibitors are making between them $9 billion a year. Here we have a protein, an antibody that can block TNF production by targeting TOL2 in the rheumatoid joint. Again, the idea here is it's dysregulated inflammation. It's TOL2 driving this pathway in the joint and causing perhaps disease. And we're very excited, of course, in our company to follow this up. And just finally on the MAL thing. Again, I can't really describe the thrill of this. We discovered it in my lab in the year 2000, just literally 5 minutes away. MAL is essential for fighting all gram-negative bacteria, okay? So our little lab found a key protein for our immune system to fight all gram-negatives. Without MAL, you can't fight them. And evolutionary consent, this is a wonderful paper came out about a month ago. It's in chickens, frogs, fish, sea squirt, sea urchin, amphioxus. All of life seems to have a MAL version. Great thrill there to find a fundamental building block. And all their immune systems in these organisms require MAL. And then we made this discovery again. People might have heard about this. It was in the Science Gallery. We found there were 2 types of MAL in human. One is called MAL-S. And there's called MAL-L, and there's the MAL gene. And even more excitingly, we found that if you were heterozygous, which means 1 copy of each, you had lower risk of malaria, TB, pneumonia, sepsis, vaccine failure was a recent one, Behçet's disease. So the heterozygotes were protected. In other words, the MAL was just right. It wasn't too hot, and it wasn't too cold. It was in the middle, and that illustrates the immune system perfectly. If your immune system is too strong, you get disease. If it's too weak, you get disease. The place to be is in the middle, and that's why that heterozygous state was protected. So TOLs, very important for fighting infection, clear evidence that they're key for infectious diseases, and this is where the new vaccines will come from. Because the new ones under trial, they've got like bits of TB in them, or bits of malaria, or bits of HIV. They've also got a chemical that activates TOLs. Now remember that the dendritic cell has TOLs and it's being activated. If you include a TOL ligand, a TOL chemical, you might boost the vaccine, and that's what's happening amazingly. So there's one in hepatitis A, hepatitis B, GSK have one in malaria. There's also one in lung cancer. All of these have chemicals that activate the TOL pathway to get the immune system going, to allow the vaccine to work. So we're very hopeful now that activating these pathways will give rise to new vaccines. Now, the last example, if you can stand it, is diabetes, right? And this is our most recent stuff. Again, part of my second program with SFI, and this is part of the Immunology Research Centre, the IRC, which is a cluster of labs in immunology in Trinity. Kingston Mills is our director. There's 9 labs in total, I think, and we're all working together. This discovery came out of that network, right? And it's diabetes, type 2 diabetes. And really, it's a very exciting story, if I say so myself. Because we may have stumbled into a cause of type 2 diabetes by accident. Now, I have no interest in diabetes. I got here again, like all great research, you're faffing around in a tool shed somewhere. And you stumble into it by accident. Now, it turns out that type 2 diabetes, now remember, diabetes, 2 types of diabetes. Type 1 means your immune system attacks your pancreas and you get no insulin. So you need to inject insulin. Type 2 is different. There's loads of insulin, but your body's not responding to it. It's called insulin resistant. Okay, so type 2 is insulin resistant. Your body cannot respond to insulin anymore. That's what type 2 diabetes is. And amazingly, about 5 or 6 years ago, they found a role for the immune system in type 2. And in particular, this thing called IL-1. Now, IL-1, remember, is another messenger molecule of the immune system. It's made during infection, key for fighting infection, just like TNF. They're both very important. And lo and behold, here it was in type 2 diabetes. Real mystery. Why would you have this immune system protein in type 2 diabetes? And what was very exciting was how IL-1 gets made was worked on in various organisms. And it's made by a multi-protein complex called the Inflammasome, which is a great name, and is a very important protein called NLRP3. That protein gives rise to IL-1 production, discovered about 7-8 years ago now. And it's especially activated by products of damaged tissue. Okay? So noxious bits of bacteria, bits of viruses, but also bits of damaged tissue activate NALP3, and you get IL-1 being made. The job of IL-1 is to wake up the immune system, but it's also to cause inflammation, to restore homeostasis, right? And things like uric acid in gout, for instance, beta amyloid in Alzheimer's disease is sensed by NALP3. So NALP3's job is to sense noxious substances. And we wondered, What would activate NALB3 in type 2 diabetes? And Seth in our lab had a great idea. He turned to this stuff. So here's a thing called IAPP. In type 2 diabetes, this stuff gets deposited in your pancreas. It's called islet-associated polypeptide. And it's a horribly noxious substance. There's the IAPP fibrils. It gets deposited. Although this is a human sample now of a type 2 diabetic after death. The pancreas is full of this IAPP. Now, because I'd seen and Seth had spotted this, that NALB3 senses noxious substances, maybe it's sensing IAPP. And that's what we discovered. We did the experiment. And lo and behold, here's IL-1 being made. And here's IAPP driving loads of IL-1 very nicely. These are two controls. We know these could activate already. There's uric acid, actually. There's IAPP. And look at this. We could take a knockout mouse that lacked NALB3, no IL-1. And that proved then that this NALB3 was sensing IAPP. Even better, we got a mouse from a lab in Washington where they put the human IAPP gene into a mouse. Why would you do that? The mouse hasn't got IAPP, strangely. And that mouse gets diabetes, a model of diabetes. And when you look at that mouse, you get loads, these are the beta cells all dying in the pancreas, and loads of IL-1 is being made in that mouse. It's a transgenic mouse, so we overexpress the IAPP. And you can do this co-localization, yellow, like the confocal. The yellow is IL-1 beta being sensed with the amyloid, the IAPP in red. They co-localize. And they're in macrophages, the key immune. So they can stain the cells for macrophages, or the tissue, MAMA2, these little red dots. Here's IL-1. The yellow, if you can see it, is co-localization. So basically, in vivo, that is, we could show IAPP is driving IL-1. What does this mean for diabetes? Here is one idea as to what causes type 2 diabetes. I call this 6 steps to hell. It normally starts with obesity and low-grade inflammation. Irritation in the body begins to sort of take its effect. And for some reason, cells stop responding to insulin because of this low-grade inflammation, probably caused by obesity as a noxious event in your body. What happens, of course, is the beta cells make more insulin to compensate. They also make IAPP. And IAPP is a horrific thing because once it begins to build up in concentration, it forms these horrible noxious clumps. They're called oligomers. And what happens then is the immune system doesn't like those. So the macrophage goes in. NALB3 senses IAPP. IL-1 is now made. IL-1 is a potent pro-inflammatory factor. It leads to further insulin resistance. It actually kills the beta cells. So this is really horrible. It makes the whole thing worse. And then finally we get type 2 diabetes. So NALB3 is a key driver of type 2 diabetes. That would make it a very good drug target. And of course, that's the one thing we're trying to follow up on in Opsona. And we thought this was great, you know, and we're very happy with ourselves. And then a drug company in California last week published a trial blocking IL-1, and it didn't work. A company called Zelma. They did an anti-IL-1 antibody, and they did a phase 2b looking at glucose. No effect, right? They did see effects on lipids and things. That took the wind out of our sails slightly, right? Now, why the trial failed, we don't know. It's a common tale in the industry. It could be a different form of IL-1, actually, IL-1 alpha. Might be important here, but still, you've got to watch the hype, I suppose, is the way to think of it. And then it's not just diabetes. All of these diseases have noxious substances irritating the immune system. And that's why we think the immune system is really going out of control in these diseases. So things like atherosclerosis is cholesterol crystals, Alzheimer's is beta amyloid, a thing called AMD has drusen's, Huntington's has something, Parkinson's has Parkin, motor neuron disease, SOD1. All of these drive the immune system crazy through NALB3 and IL-1. And that wasn't possible to say about a year ago. That was discovered in the past year, that all these things are going through these pathways. And again, my optimism to say we're getting close now to understanding these diseases in terms of how they work and how they might be treated. So where does that leave us? Well, as I've said, the prospects are very good. You've got excellent prospects for new treatments, for inflammatory disease, new vaccines and cancer. All of that is a reality. Massive investment in the pharma sector in all these, and the smaller biotech sector. And it's very exciting. The brightest and best are going into these companies to exploit these findings. And then also us academics are in our labs trying to make discoveries that could be relevant for these diseases. Many drugs in development, about 100 are in development at the moment for all these diseases. And some of the trials looking very good. There's always little trip-ups, of course, and so on. And most importantly of all, I think, we're getting closer to understanding causes. It'll be environment, things like infection, perhaps, or diet. But very importantly, genetics is a key part of this. Not everybody gets diabetes. There must be some genetic background in the immune system that makes it dysfunctional. The example I gave was mal. The wrong type of mal, you have a higher risk of malaria because your immune system isn't working, right? There'll be loads of genetic differences that we're still uncovering in the immune system to explain why some people get these diseases and some people don't. And look at this, death rate at all-time low. This was in the Daily Express. And that was last summer, right? That's mainly because of statins. Where's statins? I mentioned up here, statins, yeah. There's been a giant leap for mankind realizing that cardiovascular disease is inflammatory. Statins are anti-inflammatory. So this could come true, basically. The death rate may continue to fall, and we'll have treatments for all these diseases. But let me finish with just one or two quick things. So whenever I give a talk in public, or I'm in the newspaper, not on the court case pages either, I always get letters from the general public. And there's a huge amount of suffering out there, and this is not to be overstated, how much people are desperate for new children diagnosed with bad rheumatoid. The parent would email me and ask me for help. And I've got to say, I'm not a doctor, I'm just a scientist here, and I refer them on to mates of mine and James's or whatever. So, there's a serious bit to this, because look at this. Now, mind you get some great letters as well. This one came in about 3 months ago. I gather you found a cure for arthritis, which is wonderful news. And I wonder if you have any information you could send me, as I do not relish ending up my days in a wheelchair. I can still fish for salmon once a fortnight. If ever you are in, I won't tell you where it is, just to keep it anonymous. Come down and stay, I'll take you fishing. Isn't that nice? So I thought about going fishing. But this second one you get some weird ones as well. Look at this one. Dear Professor ONeill, I am enclosing some samples for you to examine. I have been suffering from a very severe pain in my arm for the past year. What is peculiar is that it occurs at precisely the same time, times 5 p.m. and 1 p.m. I wonder, I would be grateful if you would look at these scabs under the microscope. And in the envelope, scabs. I decided to sort of send those off to some archive somewhere. But that's what you get when you're in the news. Maybe we could find a cure for some disease in those scabs. But as I say, there's such a need for information. And it's good to see so many people, I suppose, because there's a huge hunger for information and, of course, a desperate need for new treatments. And the message has to be optimistic, that these understandings will help us. And remember, the prize I have on this really here, if we can understand the immune system, we'll find treatments for all these diseases. That's the prize. Have to apply science, of course, not quackery or nonsense, to understand the immune system, and that really is the big hope. And I'll finish on that and thank the people in the lab. So this is my lab at the moment, and the data I showed you was Gavin down here, the fourth year. Jennifer Dowling helped him, she's a postdoc in my lab. Seth did all the work on IAPP, and the rest of the lab, of course, helped in various ways. But let me finish with one last thing. From that book of, there's SFI, by the way. Sorry, Graham, I meant to thank SFI as well. Very important collaborators, actually. That diabetes story, there were like 7 labs around the world collaborating with us. We were lead lab. They're all collaborating with us. Big effort. It's a huge enterprise, this. But let me finish with this. This is my favourite thing from that book about stupid answers. Maths, right? There are 300 students in Year 10. Mary and Mark want to find out Year 10's favourite colour. Mary asks 30 people. Mark asks 150 people. Mark says, my conclusions are more likely to be reliable than Mary's. Why does Mark think he's right? And some student wrote down, because Mark is a man. Thank you very much for...
Very much for that. I enjoyed it. I'm sure everybody else did.
Well, you laughed in all the right places.
Yeah,
thanks. Nice.
you prepared me. I did a little wink. So like Graham said, you talk in this very accessible and engaging way. So we'll just have a bit of a chat, like I said, and I am going to pretty soon start asking you guys for questions. But I know, so I'll give you a few minutes to think about it. But I was very happy to see you mentioning genetics in the end.
Very important.
I'm a geneticist, so yeah, it's really important. But so just one thing though, when you're talking about cancer. So cancer, we use one word for it, but it's not really one disease. It's a complex of many different types of things. So, and so when you say a cure for cancer, you mean the cures for the cancers, I suppose.
That's right. Well, there are common, as you know, there's common features. I mean, tumour suppressor genes are often shared among 10 types of cancer. So there's no doubt there's a common pathway that gives rise to cancer, and it's the cell type, I suppose. So some are leukemias in the immune system, some are in the brain, glioblastoma, So it depends on the tissue, I suppose. But broadly speaking, the main pathways are shared. So the anti-cancer drugs then could have wider utility. One very good example is a tumour has to get a blood vessel system to grow. It's called angiogenesis, and it attracts in new blood vessels. And then it gets a blood supply, basically. There's a drug called Avastin, which stops that. It works in about 4 different types of tumours.
Thalidomide as well, right?
Thalidomide works in
which
cancer
is all the
as well.
bad press for, of course, when it was a morning sickness drug, was causing these birth defects, which were because there wasn't proper vascularization in the embryo, but other times we want to stop the vascularization.
And that blocks angiogenesis as well. So, I mean, those drugs are working in, like, the big ones are the big solid tumours like lung. They're still very difficult to treat. But these newer drugs are emerging now that may work in these ones as well. And like the example I gave of that ipilimumab, which is an anti-CTLA4 antibody for the immunologists in the audience, right? That activates T cells, works in melanoma. It got approved last week by the FDA. That'll be the first one, because that's just activating T cells. Now, a T cell can target any tumour. So you might have a general treatment there for many types of cancer, I suppose. So even though there are differences, of course, here and there are common pathways, I guess, is the answer.
Yeah. But another thing, so, in the beginning you mentioned, was it Hunter who was talking about inflammation is good, except when it's not. You're talking about drugs that will block various components of the immune system. So presumably you don't want to completely shut these off because they have their uses as well, or is that, in certain circumstances, a price you're willing to pay? Would you completely shut this down?
Are you one of our investors in our company? Keep asking me that. Why would we give you money? You're going to kill someone. There is always a risk of infection. Always a risk. And people on one of the anti-TNF drugs, infliximab, they get TB. So in other words, these systems are there to protect us from infection mainly, right? So if you tamper with them, you'll have a risk of infection. And that seems to be manageable. So you can come up with ways of watching the infection emerge. So before you go on that drug, they test you for TB, for instance. Another bad example people may have heard of is Elan, our very own Elan, discovered a treatment for MS called Tysabri. That stops T cells going into the brain. Gave it to patients, and some of them died of a virus called JC virus, which came out of their brains, right? Now, again, they withdrew the drug. Now they've got tests for that, and they can treat it. So there are ways of hopefully handling this risk
so you have some pre-screening for eligibility,
pre-screening,
I suppose.
but it's still a worry. I mean, and some people really worry. The closer you get to the real action, the more likely you'll have immunosuppression and you may get infections.
But do you think you can get closer? So if you're talking about, though, in these diseases, it's the immune system kind of having an inappropriate reaction.
Yeah.
Do you think you can, rather than shutting down the immune system, you can get it closer to the homeostatic level? That would be a long-term goal then.
Who wrote your script, Eva? That's exactly So it's a bit like a volume control. It really is like that. And the drugs that are, like the anti-TNF, they don't block TNF completely. They crank it down a bit and leave a bit behind, basically. And that seems to reset the system into a homeostatic state. And now it goes into remission, is the idea. So too much drug is a bad thing, basically. As in most drugs, too much is bad. So just enough, seems to be the secret. And that's all about dosing. Now maybe in that diabetes trial they got the dose wrong. See, this is the thing. You don't know what they're shooting in the dark with the doses.
And sometimes that can happen, can't it? When something comes out of the lab and there are all these stages to go through, like all the testing in animals, which might seem unsavory to some people, but it's necessary. And part of the reason you do this is because one of the things you need to figure out along the way is dosing. But then you have people sending you letters who want the cure tomorrow.
Indeed.
And so there's a pressure, I think, to hurry these stages up, but you can scupper the whole project.
It's still a mystery to me. So I'll give you an example of that. If anybody wants to get a job in a drug company, it's not a great vista for the moment because Pfizer, the world's biggest drug company, let 3,500 people go on research. They shut down this place in Sandwich in Kent. There was 2,500 people. Some of my mates worked there, they were fired. 1,000 in the US in research. Terrifying. And their stock price went up. That just shows you how stupid the industry is. They were spending $1 billion a year on research and not getting any drugs. And you wonder why. What are they playing at? $1 billion a year, no drugs. And the reason is because they maybe over-rely on some of the animal models. They do a phase 1 and it works a bit and they sort of doesn't work as effectively, but they're pushed into a phase 2. So there's all kinds of reasons why drugs fail, which is the downside. And they're going to use, interestingly for Ireland, the talk is they're going to use this money they save to fund academics.
Okay.
Because academics are more likely to come up with something interesting than someone working in Sandwich in Kent as part of a big...
Will they try and determine your research agenda then?
That's the other aspect of this.
Because if you let academics do what they do well, it's by actually letting them follow the lead that they find interesting rather than the one
you don't want to ruin
the CEO finds interesting.
You don't want to ruin it. So no, well, I mean, they've realised that doing research in big, huge factories doesn't work. And of course, Viagra. So I was in Istanbul on Thursday.
Where's this going, Luke?
So Pfizer have a new drug for arthritis. It's a JAK3 inhibitor, for those of you who want to know about JAK3. And no doctor has ever heard of JAK3, because most doctors are stupid. So Pfizer flew 1,500 rheumatologists and dermatologists to Istanbul. I gave a 1-hour talk on inflammation, just to make it look kosher, right? The Pfizer guy gave a talk on JAK3, right? And at dinner that night, I was sitting beside, I was telling Graham this, I sit down for dinner at a big posh restaurant, and my name is on the thing. And this American sits beside me, and I say, so do you work for Pfizer? And they all start grinning. He's the head of marketing for all of Pfizer, this fella. It's like asking Bill Gates, have you heard about computer? And this guy said he's in charge of all marketing. They make $11 billion a year, Pfizer, on 9 products, right? Viagra is a third of that.
Wow.
Imagine that. But there was no drug like Viagra before. They revealed this market. So about $3 billion on Viagra, about $4 billion on anti-TNF. It's amazing.
And of course, it was also an accidental
And the point
discovery.
of how it build up to the Viagra was discovered
Sorry.
by accident. Yeah, which is ridiculous.
Yeah.
Isn't it? You know,
Yeah,
so,
yeah.
so it's impossible to
An
predict.
embarrassing side effect.
Yeah. Right.
Are there any questions from the audience? Anybody there who'd like to get involved? Here, we've got one here close to the front. Yeah. There's a microphone, I think, if you just wait a second.
I'm not sure if the microphone's working.
Yeah.
Okay.
You're a doctor. Rick. Hang on a minute.
I was just going to say I'm one of those stupid
I
guys.
apologise. I always like to offend people.
But I have a question that I hope you can shed some light on. Allergy,
Yes.
lots of signals. You probably also have heard of the Barker hypothesis
Yeah.
and the link between perhaps antibiotics in children and increasing allergy.
Yeah.
Do you think there's any room for manipulation of the immune system during the evolution of the immune system in childhood that might lead to better outcomes in adult life?
That's a good point. I mean, there's always reluctance, Nick, to go into kids, with new agents because there's a risk of unwanted effects. So people rarely do trials on children, and they don't want to really go near them for drug discovery. But asthma and allergy could be the one place where they would. And of course, as you probably know about the hygiene hypothesis, that's in childhood. So clearly, if you can program the immune system during childhood, you can predict a lowered incidence of asthma allergy in adulthood. They don't know the mechanism, but there's evidence for that out there. And certainly with the commensals in the gut, it's where the antibiotics are probably working. They're altering the balance of commensals in some way, and then that somehow protects against allergy. So it does look as if the hygiene idea may well be correct, and it could well be in children that you'd intervene first to try to manipulate the immune system, I guess, to prevent these allergies from happening. Because really what asthma and allergy is, it's the immune system isn't educated correctly. And it turns bad on you. It's like I went to the wrong school, I suppose, during childhood. Get it to the right school, you won't get asthma and you won't get allergy. And even amazingly, they've shown women who are pregnant, who spend time on a farm, have kids who have a lower incidence of asthma.
Worms.
Was it
Worms.
Well, no,
Do you
but that's on
remember
a
that
farm.
guy who put worms into himself and found that his asthma got better?
There was worms as well, but this farm, they got a bunch of women to live on a farm for 3 months. This is true. And then followed their kids later, and there was a much lower incidence of asthma. So the women must have been picking up some germs on the farm. And that was going in utero probably and, causing a programming of the immune system. So that's a very interesting area. And of course, asthma allergy is another big indicator. I didn't mention that much. Huge activity in that, because the incidence is so high, and trying to understand that process is a massive area. And again, there's new drugs coming down the pipeline. I'll give you one other example of that. This is last summer. So we get this thing called contact dermatitis. You get a rash from a watch or a chain. Nobody ever knew why, right? Mice don't get contact dermatitis. You can rub as much nickel as you like into a mouse for perverse reasons, obviously. And the mouse does not get contact dermatitis. It comes down to one of these tolls. So toll number 4. Remember the number? Number 4 binds nickel and causes inflammation in skin. And toll number 4 has 2 histidines. Now, you may remember your chemistry, Nick. And these histidines coordinate the nickel and cause the activation. The mice have different amino acids there. And the evidence for it, they got a mouse, right? And they just changed those 2 amino acids in the mouse toll 4, right? They switched 2 alanines to 2 histidines. The mice were born and they got contact dermatitis. If ever there was a classic case of science working superbly well, that was it. And of course, that opens up the prospect of toll 4 inhibitors, blocking contact dermatitis. And the second part of that story is, of course, work in Trinity, as you know, with eczema. The genetic risk, not everybody gets rashes, there's a genetic basis, and these filaggrin proteins in skin, these barrier proteins are slightly different in some people. That allows the nickel in. And that's why you get contact dermatitis, because of the mutation in filaggrin. So again, it's a combination of the environment, a noxious substance, and some genetic background giving rise to contact dermatitis. And it identifies toll 4 firmly as the pathologic molecule, and there are toll 4 inhibitors in development that could be used.
I would probably take them because I didn't choose my parents correctly and I have contact dermatitis.
That's right. That's the bit. How do you avoid heart disease? have different parents. wasn't that the famous joke, I think?
Is it true you hear these anecdotes of like somebody who has eczema and they get some other infection and the eczema clears up? Is there kind of a prioritization in the immune system?
I haven't heard that.
You know, I only heard it anecdotally.
Is that an old wives' tale?
No, it's just anecdotes, but
right.
I don't know. It's even less
It's not
even an old one.
Another person's old wives' tale. I never heard that.
You haven't heard that? Okay. Well, just a bit, like it reminded me of it when the person gave himself worms and his asthma cleared up. But okay, there's a question up there at the back.
Whether we should all take worms is the question.
Stupid doctor.
I love
just
that.
Wondering if you've
I must apologise to you as well then.
That's okay. I don't mind being called stupid. There have been, in human cancers, well-described incidences of spontaneous regression of tumours. And among the tumour types, there would be renal carcinomas, malignant melanoma, seminoma in men. I was just wondering, had you any ideas on what mechanism might be at work there?
Yeah.
This is well documented, histologically proven, pathologically proven, and no treatment.
Yeah, that's got to be the immune system is the view there, because obviously the genetic alteration wouldn't have changed. So you'd have the tumorigenesis because of the genes. They're not going to remutate back to normal. So the body is able to handle the tumour somehow and clear it, and that's more than likely going to be the immune system. And you do find, in melanoma especially, the ones who do regress have T-cell infiltration into the tumour. You can detect that these cytotoxic T-cells are in the tumour. And that's what provoked that trial, to be honest, because they designed a drug to activate those T-cells, and it's working to some extent in melanoma. So the most likely interpretation is the immune system gets its act together. Now, because the question is, why would the immune system be asleep then, And what would trigger it to suddenly start fighting the tumour? And that's not clear. It's not clear. And some people happens, some people doesn't. So if we could understand that process of how to get the immune system mobilised, that's where the hope is, that these solid tumours will be treatable.
Anybody else around? Looking for more. Let people think. One thing I thought of while you were talking, you mentioned HIV as an example of some vaccine hope.
Yeah.
But my understanding of the problem with HIV is that it's changing so quickly. You don't even. It's not a matter. It's not that you can't educate the immune system to recognise HIV. It's that HIV is not HIV anymore. At least it's, as far as the immune system is concerned. So is there really a vaccine hope for HIV?
Well, that's a strange one. The theory always was these things change the whole time when they escape the immune system, and there's some evidence for that, really. But there's more evidence that the virus is very good at manipulating the immune system. So viruses have all kinds of ways of turning off the immune system, and HIV is the worst for this because it lives inside the immune system and very cleverly stops the defense against it. So any vaccine then is going to be up against the capacity of the virus to stop the vaccine working by manipulating the immune system. So that's probably as important as the high rate of mutation, although the high rate of mutation is part of the picture, I guess. Yeah. And in fact, the hope with these is to activate these tolls. As I mentioned, that's more innate immunity. See, if you can trigger an innate response to the virus, that may work more better than the adaptive, which would be the T-cell response. And that's why these adjuvants, they're called, in malaria and HIV are showing such promise, probably because they're bringing out the right part of the immune system, which the bug can't escape. Because bugs are very good at escaping these things, but not as good as at escaping the innate. Yeah. And that's a huge area of immunology, which has been very fruitful. How viruses are very clever. And if we can understand how a virus turns off the immune system, that will often identify a key part of the immune system never before understood.
One of my favourite examples actually is in herpes simplex, cold sore virus, because so many people have it. Yeah. And it has a gene which actually manipulates, it has a microRNA which will switch on and off human genes. So like you kiss somebody, a virus invisible to you lands on your lip and starts controlling you like a zombie. Right, exactly. So there don't seem to be, I don't think there are any more questions, but I was going to ask you one thing kind of to wrap up. So you're talking about all these things that we will be able to survive. Like we won't be dying of cancer, infection, malaria, all these things anymore. So what are we going to die of? We have to die of something. Shall we have euthanasia? We just schedule euthanasia for 130.
In the brave new world, that is the key question. What will we die of? Old age, hopefully. I don't know. But it's an ethical question, this, in many ways, because is it correct to stop these diseases? Because there's no doubt that there'll be overpopulation. So you've got to have a situation where you're stopping people breeding. That's the other thing. You can vaccinate against that as well, by the way. So I think, but there will be a big issue in Africa. And in fact, one of the worries is if you cure malaria, massive population growth there. And that's why the Gates Foundation, they're not just funding vaccines, they're also funding education. basically allow people, or whatever you want to
right?
phrase it, to decrease the birth rate through education. So it goes hand in glove, I guess, with the two.
Educated women have fewer children.
That's right.
Yeah.
So I guess that's the other way to think of it, I suppose. I mean, you can't really stop us working on these diseases and looking for cures, so these will come about.
No, I'd rather, given that I must die one day, I'd like it to be one of those peaceful in your sleep versions.
That's right.
Yeah.
Yes, but maybe euthanasia, at the age of 30.
No, too late.
Or something like that. What are the consequences of all this research?
Okay, well, thank you, Luke. That's been really entertaining, sir. A very, very good start to the SFI speaker series, and I look forward to the rest of them. And thank you again to SFI for funding this, and you coming.
Thank you.